Okay, so ladies and gentlemen, you are welcome. So we are starting the lecture right now. So don't forget, ladies and gentlemen, as we agreed, today we are going to look at citric acid cycle. So you are all welcome. So without wasting much of our time, we are going to start the lecture in the next 2 minutes. So you are welcome. I hope you can see me and you can also see the board. Please, can you see me and can you also see the board? So, ladies and gentlemen, this is where I'm going to start. Don't forget, actually, from our previous classes, we discussed that citric acid cycle is taking place in the mitochondria. So, generally, we have 4 steps of cellular respiration from what I told you last time. We have 4 steps of cellular respiration. Number 1, cellular respiration occurs in 4 major steps. Number 1, after digestion, the first step of cellular respiration is glycolysis. And what is glycolysis? Glycolysis is the breaking down of glucose to produce pyruvate. So at the end of glycolysis, we are expecting glucose to be converted into 2 molecules of pyruvate. That is one. And then the second stages, let me just write it here. Stages of cellular respiration. Stages of cellular respiration. Stages of cellular respiration. So the stages of cellular respiration, number 1, we have glycolysis. And then number 2, we have conversion. We have conversion of pyruvate. So the conversion of glucose to pyruvate. And then number 3, Krebs cycle. Number 3, we have Krebs cycle. And then number 4, we have electron transport chain. We have electron transport chain and oxidative phosphorylation and oxidative phosphorylation. Oxidative phosphorylation. So, ladies and gentlemen, okay, good evening, sir. Sorry, good evening, Victoria. Please, can you see the board and can you see me clearly? Please, can you hear me and can you also see the board? Hey, hello, Victoria. Can you hear me and can you also see the board? So after this, the next thing, so these are the 4 stages of cellular respiration. And I think we discussed on the different aspects of glycolysis. Glycolysis occur in the cytoplasm. So therefore, pyruvate are generated in the cytosol. So remember, actually, I think for some of you that attended our last lecture, after this pyruvate is generated in the cytoplasm, but of course, citric acid cycle occur in the mitochondria. So therefore, the pyruvate generated in the cytoplasm, it must be transported mitochondria because that is where exactly citric acid cycle occur. So please put this at the back of your mind that the Krebs cycle is taking place in the mitochondrial matrix. So as I said, we have 4 stages of uh, cellular respiration. So today we are going to look at the conversion of pyruvate. Yes, here is conversion of pyruvate to acetyl-CoA to acetyl-CoA. That is the second step. So after glycolysis, the pyruvate will be generated in the cytoplasm, and this pyruvate will be transported to the mitochondrial matrix. And of course, in the mitochondrial matrix, the pyruvate will be converted into acetyl-CoA. So the question is, how this pyruvate generated as a result of the action of 5 coenzymes. There are 5 coenzymes and 3 enzymes that help the pyruvate to be converted into acetyl-CoA. So now let's look at the enzyme. So the enzyme that do this job, we call it pyruvate dehydrogenase complex. We call the enzymes that convert pyruvate to acetyl-CoA, we call the enzymes pyruvate. We call the enzymes pyruvate dehydrogenase complex. That is the name of the enzymes. The name of the enzymes is called pyruvate dehydrogenase complex. So that is the name of the enzymes that convert pyruvate to acetyl-CoA. We call it pyruvate dehydrogenase complex. It is called a complex because it encompasses of 3 enzymes in it. So in biochemistry or in enzymology, when we said enzymes is a complex, it means that it has other enzymes in it. It means that it is not a single enzymes. Please, are you seeing the board or you are having problem with the board? Yes? Are you seeing the enzymes? Or sorry, are you seeing the board or you have problem? Because I want to know, is it that the problem is actually coming from you? So as I said, it is enzymes, and this complex includes enzymes E1, enzymes E2, and enzymes E3. So enzymes E1 under this complex comprises of 3 enzymes in it. And the first enzyme is called pyruvate. The first enzyme is called pyruvate dehydrogenase. Dehydrogenase. That is the name of the first complex. And then the second enzymes under this complex is called transacetylase. The second enzyme is called dihydrolefoil dihydrolefoil transacetylase. So that is the name of the second enzymes. And then the third enzymes is called dihydrolefoil. The third enzymes in this complex is called dihydrolefoil dehydrogenase. It's called dihydrolefoil dehydrogenase. So I want you to understand, ladies and gentlemen, you should never forget that when we are talking about the combustion of pyruvate to acetyl-CoA, this combustion is done by a complex enzyme that it is called pyruvate dehydrogenase complex, and it is called complex because it encompasses of three enzymes in it. And these three enzymes, number one, we have pyruvate dehydrogenase. Number two, we have dihydrolefoil transacetylase. And then number three, we have dihydrolefoil dehydrogenase. So these are the three enzymes that are under pyruvate dehydrogenase complex. And very importantly, that you should never forget, ladies and gentlemen, as I said, okay, the board is not clear. Okay. Let me see if I can do something. Okay, give me some minutes so that I can make it more clearer. Please, is the board clear now or I should? Is the board clear now? Okay. So this is the complex. And don't forget, I told you under this complex, there are, apart from the three enzymes, and it also encompasses 5 coenzymes. And what are those 5 coenzymes? Number one, we have FAD plus, we have NAD plus, we have lipoids. We have lipoids. That is number 3. And then apart from lipoids, there is also TPP. TPP. That is thymine pyrophosphate. Thymine pyrophosphate. And then the last one is coenzyme. Is CoAsh. So these are the 5 coenzymes that required by pyruvate dehydrogenase complex to convert pyruvate to acetyl-CoA. So that is what I want you to understand. So let me repeat myself for those of you that just joined. That after glycolysis, two molecules of pyruvate are generated. And those two molecules of pyruvates that are generated in the cytoplasm. They are generated in the cytoplasm and of course or in the cytosol. So Krebs cycle occur in the mitochondria. So therefore, the pyruvate must be transported to the mitochondrial matrix. And in the mitochondrial matrix, there is an enzyme called pyruvate dehydrogenase complex. And this pyruvate dehydrogenase complex is the enzymes that convert the pyruvate to acetyl-CoA. Because the starting material for Krebs cycle is acetyl-CoA, so you need acetyl-CoA. It is a major fuel that initiate Krebs cycle. So that is what I want you to understand. And this pyruvate complex, it is called a complex because there are so many enzymes under it, and that is why it is called a complex. Complex is like a normal English. When we said complex, it means that it encompasses so many things. So this one is called pyruvate complex because it encompasses of three different enzymes in it. These three enzymes we have pyruvate dehydrogenase, we have dihydrolyfoyl transacetylase, and then we have dihydrolyfoyl dehydrogenase. So these are the three complex enzymes. So after that, you generate acetyl-CoA, and of course, these coenzymes they also generate, of course, as they enter like this, they also produce a product. Like this NAD plus will produce NADH. The FAD will produce FADH2. It will be generated. So these are some of this. Of course, it is this complex. It encompasses of 5 coenzymes. Please don't forget there are 5 coenzymes: TPP, thymine pyrophosphate, CoASH, FADH, NAD plus, and then lipoil. So these are the 5 coenzymes that are required by this enzyme. So that is what I want you to understand. So then the next thing, after you generate acetyl-CoA, what happened? This acetyl-CoA, as you see it from here, pyruvate is a 3-carbon compound. Pyruvate is a 3-carbon compound. And then acetyl-CoA is a 2-carbon compound. So this oxaloacetate and acetyl-CoA will condense themselves. They will join together. 2 molecules, oxaloacetate and acetyl-CoA, will combine themselves together. They will combine themselves together. So once they combine themselves, what they will generate? They will generate what you call straight. So straights will be generated, as you can see from here. When you have acetyl-CoA react, as you can see from here, this is oxaloacetate. From here, this is oxaloacetate. So the acetyl-CoA and oxaloacetate will combine together to form straights. And the enzymes that do this work, that convert these 2 molecules together, is called straight synthase. It's called straight synthase. So that is the name of the enzymes. The enzyme straight synthase convert oxaloacetate and acetyl-CoA, condense them together to produce what you call straights. So after this straight is generated, what next? The citrate will be converted to generate cis straights using an enzyme called aconitase. So aconitase is an enzyme that remove a water molecules. So it removes water molecules. So one molecules of water from here will be released or will be removed from citrates to produce cis strates. And the enzyme that do this work is called Aconites. Aconites. Let me use red marker to show difference. We call it aconites. So aconites is the enzymes that convert I hope you are following. So aconites is the enzyme that convert straight to cis straight by removal of one molecule of water. Please, are you following? Please, are you following? Do you understand up to this stage on how first acetyl-CoA generated, acetyl-CoA oxaloacetate condensed to produce straight, and then straight using an enzyme called straight synthase, and then the straight will be converted to cis straight. And the enzyme that do this work is called aconites. And what this aconites does, yes, excellent. I am happy that you are following. So I benefit myself. Citrate is produced. Then another guy or another enzymes called aconites will now come and see that, okay, in the citrate there is an extra water molecules that is not needed, so it need to be removed. So the enzymes called aconites will remove one molecule of water at this point. So once this one molecule of water is produced, a cis isomer is generated in the citrate. So that is why it is called cis. It's called cis. Where did you confuse, please? Where did you confuse? Is it Jessica? Where did you confuse, please? I don't want you to confuse. I'm just taking it bit by bit so that for all of you to understand. Yes. Okay. So the next thing, after one molecules of water is removed to produce cis straight, the next enzymes is also another aconites. Will say that there are still extra molecule of water and we have to remove it. So then another molecules of water here will also be removed. Another molecules of water here will also be removed. So another molecules of water will be removed from here. So you see we have two aconites. So then what next? Once another molecules of water is removed from aconites, from cis straight, then iso straight is generated. Iso straight is generated. So what next? After this isostrate, then this isostrate will be converted to alpha ketoglutarate. Please, if you are going to forget anything in Krebs cycle, you should never forget this step. Please jot it down. If you are going to forget any step in Krebs cycle, so you should never, there are some key steps that I'm going to mention that you should never forget them. So this isostrate will be converted to alpha ketoglutarate. And this steps is very, very important. Why? At this point, an enzyme called isocitrate dehydrogenase will work. Isocitrate dehydrogenase. It is the enzymes that convert isostrate to alpha ketoglutarate. And why did I say this step is very important? Because at this step, NADH is generated. So NAD plus. Let me be using blue marker for products that are released. So NAD plus will be converted into NADH plus H. So that is why I said you should never forget this step. Isostrate dehydrogenase will convert isocitrate to alpha ketoglutarate. And this step is very important. And why did I say this step is very important? Because at this point, NADH is generated. And at the end of this class, I will tell you the importance of this NADH. So what next? After alpha ketoglutarate is generated, then the next thing, the alpha ketoglutarate will be converted into acetyl-CoA. And the enzymes that do this job, what happened actually at this step? What happened? Isocitrate will be converted to alpha ketoglutarate. Then, so what happened? The alpha ketoglutarate will be converted into acetyl-CoA using an enzyme called, look at it, the name of the enzymes that convert alpha ketoglutarate to acetyl-CoA is called alpha ketoglutarate dehydrogenase. So you see, for this isostrate dehydrogenase, I told you that this step is very important. But do you know that the conversion of alpha ketoglutarate to succinyl-CoA is also very important, even at some point more important than this? It's not that they are not all important, but I will tell you why this one is important. This is one of the questions. This is one of the questions that usually asked in the DLC examination. They ask a lot of questions at this step, and I will give you the questions and then provide the answer in case when you come across such questions in your subsequent examination. Why is it that this step is important? This alpha ketoglutarate is an enzyme that at the same time also convert NAD plus to NADH plus H. And also at the same time, one molecule of carbon dioxide is removed from here. So one molecule of carbon dioxide is removed from alpha ketoglutarate to succinyl-CoA. So one molecule of CO2 is removed. So therefore, alpha ketoglutarate will be converted into succinyl-CoA. And of course, at this stage, one molecule of NADH is generated, and then one molecule of carbon monoxide. Is that clear? So, are you following, please? Do you understand this stage? But I want to tell you that it's something very important apart from this conversion. For this alpha ketoglutarate dehydrogenase to work, it needs an enzyme. It needs coenzyme. And the coenzymes that these enzymes need is TPP, thymine pyrophosphate. So now, what are the questions that I usually ask? Thymine is which vitamin? Which vitamin is thymine? That is the question. Which vitamin is thymine? Which vitamin is thymine? That is the questions that they usually ask in the exam, but I want to start asking you, which vitamin is thymine? Is it vitamin A? Is it vitamin B? Is it vitamin C? Is it vitamin D? Is it vitamin K? If it is vitamin B, B is a complex which include vitamin A1, B1, B2, and B3, B12. B9, and so many. Enzymes, they are molecules that speed up the rate of the chemical reaction, while coenzymes, they are biomolecules that use by the enzymes. So it is required by the enzymes for them to combat. Okay, let me shift this small so that you can see. Okay. Excellent. Is vitamin B1. So do you know the question that they usually ask? And I want you to jot it down. What is the relationship between carbohydrate metabolism and beriberi? What is the relationship between carbohydrate metabolism and beriberi? That is the question. They usually ask this question in Biochem 242 examination. So I want to ask the question and then we answer it here so that at least when you come to such questions, you can easily address it. So vitamin B1 is thiamine. So the question is, what is the relationship between beriberi and carbohydrate metabolism? Beriberi is a disease that is associated with deficiency of vitamin B, and it is energy metabolism. It affects energy metabolism. So now how? As you can see, if there is deficiency of, as I said, somebody asked one beautiful questions. That is Eunice. I like the question you asked. She said, what is the importance of, what are the differences between enzymes and coenzymes? So let me actually differentiate it. I think if you could remember, especially those that I took by Chem 241, we have what we call we have what we call APO enzyme. APO, we have what we call APO enzyme. So when we said APO enzymes, you can call them APO enzymes or pro enzyme. So APO enzymes or pro enzyme, it is inactive form of enzymes. The enzyme is inactive. It is the inactive form of the enzyme. So the enzymes cannot work the way it is like this until when there is a coenzyme. So APO enzymes plus coenzyme, it will give you what you call a holo enzyme. Holo enzyme. So what is holo enzyme? Is an active enzyme. So what does that mean? It means that enzymes cannot work without coenzymes. So the combine of APO enzymes and coenzymes is what will give you active enzyme. So that is to tell you, I think at this point, as you can see, I told you that this alpha ketoglutarate, it needs the TPP, that is thymine pyrophosphate. So thymine pyrophosphate, TPP, thymine pyrophosphate, it is a coenzymes coming from vitamin. It is vitamin B1. So now if there is deficiency, of vitamin deficiency, that is why I cross it. It means that if there is absence of vitamin B1, that is deficiency of vitamin B1. So the question now is that, do you think that the alpha ketoglutarate will be converted into succinyl-CoA? That is the question. Do you think that alpha ketoglutarate will be converted into succinyl-CoA if there is deficiency of vitamin V1 in the body of an individual? That is the question. And I need answer. Do you think that alpha ketoglutarate will be converted into succinyl-CoA? If there is deficiency of vitamin B1, it means that there is no TPP. So if there is no TPP, do you think alpha ketoglutarate will convert, alpha ketoglutarate dehydrogenase will convert alpha ketoglutarate to succinyl-CoA? That is the question. I'm waiting for the answer. Excellent. Zalihat and Halimat Saif, the answer is no. So there would be no combustion of alpha ketoglutarate to succinyl-CoA. So if there is no that combustion, do you know the implication? Even this NADH will not be generated. So therefore, the cycle will completely halt. It will halt from this stage of alpha ketoglutarate. So the alpha ketoglutarate will be accumulated. Even the isostrate, it will be there at the end. There will be colossal accumulation of alpha ketoglutarate and isocytrate dehydrogenase, and that will lead to a lot of disease like kidney disease, inflammation, with a lot of complication. Excellent, because it requires coenzymes for the combustion. Excellent. And this NADH you are seeing, they are coenzymes that are converted into ATP. So that is why beriberi, that is vitamin B1 deficiency, it linked with beriberi. And beriberi is a disease that associates with a low energy. There is usually low energy in an individual with beriberi. Beriberi, yes. Because this NADH and ATP cannot be generated. And if there is no ATP, it means no energy. So I'm very happy that you understand that. So let me continue. So you see here we have succinyl-CoA. So what happened? Oh, you confused from isostrate. So let me repeat myself. Idara Friday. Isostrate is produced by the combustion of cis straight to isostrate using an enzyme called aconitase, and one molecule of water is removed at this point. After we have this isostrate, so isostrate will be converted into alpha ketoglutarate. And the enzyme that convert isostrate to alpha ketoglutarate is called isostrate dehydrogenase. And at this point, NADH is generated, which is very important. And then, of course, from this, the alpha ketoglutarate will be converted into succinyl-CoA using an enzyme called alpha ketoglutarate dehydrogenase. And this enzyme, alpha ketoglutarate, it requires TPP, that is thymine pyrophosphate, as a coenzyme which convert also one molecules of NAD plus to NADH with the removal of carbon dioxide. So it means that at the same time, these enzymes generate NADH with one molecules of carbon dioxide. Is that clear now, Idara? So I now said, what is TPP? TPP means thymine pyrophosphate, which is the same as vitamin B1. So if there is deficiency of vitamin B1, what will happen? It means that there is deficiency of TPP. And if there is deficiency of thymine pyrophosphate, so the alpha ketoglutarate dehydrogenase cannot convert alpha ketoglutarate to succinyl-CoA. So that is what I'm saying. That is the implication of deficiency of TPP. So, and I said that there is one question that they usually ask: the relationship between beriberi and carbohydrate metabolism. So this is carbohydrate metabolism that they are talking about, and specifically the conversion of alpha ketoglutarate to succinyl-CoA. In fact, I think if you can see from here, from the very first step, ladies and gentlemen, look at it. I say that in the first steps we have also TPP. So if there is no TPP also at the same time, even at this stage, the pyruvate cannot be converted into acetyl-CoA. Although there are other sources of acetyl-CoA. So it can continue with the citric acid cycle, but it cannot cross this step. That is the conversion of alpha ketoglutarate to succinyl-CoA. Because we can get acetyl-CoA from fatty acid. We can get acetyl-CoA from ketone bodies. Because the sources of acetyl-CoA is not just only from pyruvate, there are also other sources. So that is what I want you to understand. So the next thing, of course, is the conversion of succinyl-CoA to succinate. The conversion of succinyl-CoA to succinate, and the enzymes that do this work is called succinyl-CoA synthetase. It's called This is the enzyme. It's called succinyl CoA. Succinyl CoA synthetase. Synthetase. That is the name of the enzymes. That is the name of the enzyme. It's called succinyl-CoA synthetase. And this enzyme succinyl-CoA synthetase, it is also very important in conversion of GDP plus inorganic phosphate to produce GTP. So this is also another very important step in citric acid cycle. GDP plus inorganic phosphate combine to produce GTP. And also, at the same time, at this point, coenzymes is also removed. CoAsh. So let me delete this so that let me delete this. So then the next thing, ladies and gentlemen, is that succinyl-CoA will be converted into fumarate. Succinyl-CoA will be converted into fumarate using an enzyme called succinic or succinyl-CoA succinate dehydrogenase. You can call it. The enzyme is called succinate dehydrogenase. Succinate dehydrogenase. So that is the name of the enzymes that convert succinate to fumarate. But I want to say something also here, very important, that you should never forget about it, because that is part of the things that we are going to do when we come to discuss electron transport chain. So at this point, the conversion of succinyl-CoA succinate to fumarate, it requires the work of an electron mobile carrier, ubiquinone Q. We call it ubiquinone. So oxidized ubiquinone will be converted into reduced ubiquinone, that is QH2. The Q stands for ubiquinone. The Q stands for ubiquinone. So it means that if you can look at it from here, ubiquinone is oxidized. It means that, look at it, there is no any hydrogen when it enters the cycle. And then, of course, 2 molecules of hydrogen is removed from succinate and loaded on the ubiquinone. That is why we have reduced ubiquinone QH2. So it produces a fumarate. And then the next thing is the conversion of fumarate to malate. Conversion of fumarate to malate using an enzyme called fumarase. Let me use red marker. Using an enzyme called fumarase. So we have fumarase. So after this fumarase, what next? The malate will be converted into oxaloacetate. So which enzyme do it work? The enzyme is called malate dehydrogenase. Dehydrogenase. And also at this point, these enzymes, malate dehydrogenase, also at the same time, it also use NAD plus. Let me use blue marker for you to understand. NAD plus to generate another NADH plus H. So now, please. I'm now going to ask you a series of questions after finishing this. First, how many pyruvate are generated in the electron? How many pyruvate are generated after glycolysis? How many molecules of pyruvate are generated after glycolysis? How many molecules of acetyl-CoA are generated after glycolysis? How many pyruvate? Pyruvate. Excellent. Excellent, Hafiz. So 2 molecules of pyruvate are generated. So if you generate 2 molecules of pyruvate at the end of glycolysis, so how many acetyl-CoA are you going to generate? During the conversion of pyruvate to acetyl-CoA, how many? How many acetyl-CoA are you going to generate? If you have 2 pyruvate here, then how many acetyl-CoA are you going to generate? Pyruvate is a 3-carbon compound. And of course, acetyl-CoA, I told you, is a 2-carbon compound. So if you have 2 pyruvate, how many molecules of acetyl-CoA are you going to generate? Yes? I am listening. I'm waiting for you. If 1 pyruvate produce 1 acetyl-CoA, so 2 pyruvate will produce or will generate 2 molecules of acetyl-CoA. If 1 molecules of acetyl-CoA can undergo 1 cycle, if 1 molecules of acetyl-CoA can undergo 1 cycle of Krebs cycle, so it means that 2 molecules of acetyl-CoA will undergo 2 cycles. Okay, no problem. I will send it. So it means that you have 2 molecules of acetyl-CoA. And these 2 molecules of acetyl-CoA, if you have 2 molecules of acetyl-CoA, then how many cycle of citric acid cycle are you going to? Okay, how many cycles is it going to occur if you have 2 acetyl-CoA? If 1 acetyl-CoA undergo 1 cycle, so 2 acetyl-CoA will undergo 2 cycles. So 1 acetyl-CoA can undergo 1 cycle. Then it means that 2 molecules of acetyl-CoA will undergo 2 cycles. That is what I want you to understand. It undergo 2 cycles. So therefore, if we have 2 acetyl-CoA, it means that at the end, each of these are 2. You have two straight. You have 2 isostrate. You have 2 isostrate here. You have 2 molecules of alpha ketoglutarate. And also, all these molecules that are generated, like here we have water. So it means that at this point, we have 2 molecules of water since we are using 2 acetyl-CoA. And also at this point, we have 2 molecules of water. And then, of course, for this NADH is also 2. For this NADH here is also 2. For this GTP, it means that you have 2 GTP. For this acetyl-CoA, it means you also have 2 NADH. So now, if you look at it, at how many steps do we generate NADH? At how many steps do you generate NADH? Please, you should be fast in answering these questions because time is going. I don't want to take too much time. At how many steps do we generate NADH? No, Saifullah Halimat, it's not 2. At how many steps do we generate NADH? 3 steps. What is the first step? The first step is where look at there is conversion of molecules of NADH is generated at this point. Then the other one, look at it at this stage also. At the point of isostrate dehydrogenase, you generate NADH. At a point of alpha ketoglutarate dehydrogenase, look at it, you also generate another NADH. Look at it very well here. You also generate another NADH. So this is 2 steps. And what is the other step? Malate dehydrogenase, you also generate another NADH. So now the question now that I want to ask after this, so how many NADH are two for two cycle of, because we're having two acetyl-CoA. So from two acetyl-CoA, how many NADH are generated for two cycles? For two cycles, how many NADH are generated? Excellent, excellent, Oluwatoyin. Six. Yes, Zalihat, six. Because you have three steps where you produce one. So if you have two cycles, it means that at the end of Krebs cycle, you have two molecules of NADH. Excellent. Then how many molecules of water are generated at the end of at the end of Krebs cycle? How many molecules of water? I'm just trying to bring out all the number of NADH that are generated. The products that are generated at the end of Krebs cycle. And ladies and gentlemen, please, I want to say something very important. Very important. These three steps where molecules of NADH are generated are the point of regulation. Are the point of regulation of glycolysis of Krebs cycle. They are the point of regulation. They are called the control point. They are called the control point of glycolysis. Of Krebs cycle. It is the step where you can control Krebs cycle. So this is also another question that you should actually put at the back of your mind. Put it at the back of your mind. How many regulatory steps do we have in Krebs cycle? We have three. What are those steps? What are those steps? Those steps are the step where we produce NADH. So alpha isostate dehydrogenase is a point of regulation. Alpha ketoglutarate dehydrogenase is a point of regulation. Then malate dehydrogenase. So now the next thing is, let's bring about, okay, now how many molecules of water are generated? Two are generated here, two are generated. So if you add them for two cycles, it means that we generate 4 molecules of water at the end of Krebs cycle. Then how many CO2, carbon dioxide? Remember, I told you that 1 molecule of carbon dioxide is generated. So therefore, for 2 cycles, we have 2 molecules of CO2. So 2 molecules of carbon dioxide is generated. Then, do we generate ATP molecules? Is ATP generated in Krebs cycle? The answer is yes, because GTP is the same thing as ATP. Please get it right. ATP is the same thing as GTP. So 2 molecules of GTP are generated in 2 cycles. So also we have GTP. So ladies and gentlemen, let me revise. In the next 4 minutes, we are going to stop. In the next 4 minutes, we are going to stop. And I want you to go through this and try to get the basic points. And please let me repeat myself. If you know that you didn't register with my YouTube channel, please try to register. And tomorrow by 8 p.m., we are going to do Human Physiology 228. Human Physiology 228 tomorrow. So let me repeat myself. At the end of glycolysis, two molecules of pyruvate are generated in the cytosol. And of course, Krebs cycle occur in the mitochondrial matrix. So therefore, the pyruvate generated in the cytosols must be transported from the cytosol to the mitochondria using a transport system. I explained this last time. Then the pyruvate will be converted into acetyl-CoA using an enzyme called pyruvate dehydrogenase complex. It is called complex because there are three important enzymes in it. That is why it is called complex. And this enzymes complex, it requires 5 different coenzymes for it to convert pyruvate to acetyl-CoA. Promise is 6 NADH, not 12. You can revisit the video. And then, of course, this complex includes three enzymes. We have E1, E2, E3. So this complex includes pyruvate dehydrogenase, dihydrolyfoyl transacetylase, dihydrolyfoyl dehydrogenase. And Krebs cycle include 8 steps. It has 8 steps. And there are 3 regulatory points. That is, we have 3 regulatory points. Is the point where you can control the pathway. And this is where any is the point of regulation. Alpha ketoglutarate dehydrogenase is the point of regulation. Then malate dehydrogenase is also another point of regulation.